This video is going to start with a quick overview of the anatomy of the human spinal cord, and then look at vertebrate evolution and look at how these structures have evolved. So first off, the spinal cord, like all of the nervous system, is highly organized. It is the most complex structure in the known universe, the human nervous system. So obviously without some degree of organization, it simply would not work. One of the first things that we see is that there is a distinction between what is called gray matter and white matter. The gray matter contains the soma, the cell bodies of neurons, while the axons are then organized in the white matter. And so there are different parts of the cells, the neurons, which compose the nervous system. And these are organized into gray and white matter in a cross-section of the spinal cord, and you can see that visibly. And the soma don't all do the same thing. There are soma which are processing sensory information. They tend to be located in the dorsal horns, and we use the term horns because in most sections of the spinal cord, if you were to describe this gray matter, it looks like a letter H, or some feel a butterfly. So the dorsal horns would contain soma processing sensory information, while the ventral horns possess the soma of the motor neurons whose axons then leave the central nervous system and say go to muscles or glands. So not only is there gray versus white matter, but within the gray matter, there is organization to form sensory dorsal horns and motor ventral horns. In between, there's a connecting section known as the gray commissure, where there is a central canal, a space through which cerebrospinal fluid passes, as we'll see. So the gray matter is organized. The white matter is as well, as we'll see in a second, but before that, so if you were to look under the microscope at the human spinal cord, you would then clearly see this has been stained, but there is a distinction between the gray matter. This is where the soma of neurons are. So here you can see the large soma of these neurons versus the white matter where the axons are located. And so once again, there is gray matter organized into both dorsal horns processing sensory information and ventral horns with the soma of the motor axons. And here you can see the white matter there. Also note that in the center, there is a hollow space through which cerebrospinal fluid will pass. This is the central canal, and a connecting section, the gray commissure. So the white matter is also organized as we will see. Oh, I'm sorry, before that. Then just lining this central canal is a type of glial cell. There is a supporting cell type in the nervous system, not in addition to neurons, known as glia, that they perform a number of roles. One set has cilia that helps to create a current in the cerebrospinal fluid. If you look at the central nervous system as it forms in embryos, here you can see the spinal cord is hollow. And if you look at the brain of this embryonic pig, notice it is hollow. So we vertebrates, here's the pig spinal cord as an embryo, have a hollow nervous system. It starts off with a space, and these spaces exist in adults as ventricles in the brain or the central canal of the spinal cord. And there's a fluid that floats through these spaces known as cerebrospinal fluid, and it is lined by a type of glial cell known as ependymal cells whose cilia help to create a current in this. So cerebrospinal fluid is made at very porous capillaries known as choroid plexus, where a filtrate from the capillaries then forms the cerebrospinal fluid. And here you can see the ciliated glial cells known as ependymal cells, which are lining the central canal. The white matter is also organized. So there are axons, but some axons are going up the spinal cord, and that's called ascending. And they obviously are carrying sensory information. So the brain doesn't know anything about what's happening on the skin, or what's being held in the hand, or muscles contracting, etc. So it has to be told about touch and pressure and pain and temperature and itch and tickle, etc. And so there are messages which then go up the spinal cord. They are ascending tracks, and notice they are organized. The ascending tracks are separate from the descending tracks. Also, within the ascending tracks, there is organization. So touch will go up the posterior column, but these yellow tracks carry touch from the hip and leg, while the red tracks carry touch information from the arm and shoulder. Here in blue are spinocerebellar tracks carrying information about body position to the cerebellum. And then there are spinothalamic tracks, which will carry pain and temperature information. So not only is the white matter organized, that's where the axons are, but the ascending tracks are separate from the descending tracks. And among the ascending tracks, pain and temperature information is going up in a different place than touch information or proprioception. That's the ability to know where your body parts are at one time. Many of these tracks, you know that they're ascending just by looking at the name. Spinothalamic, starting in the spinal cord, going up to the thalamus. Spinocerebellum, starting in the spinal cord, going up to the cerebellum. In the same way, there are descending tracks going down the spinal cord. So their names often give them away. So they will end in spinal. So corticospinal goes from the cortex to the spinal cord. Reticulospinal goes from the reticular formation to the spinal cord. So not only are descending tracks separated from the ascending tracks, but they are organized. So that's a voluntary muscle commands in the corticospinal tract are organized in a different set of spaces than the involuntary commands, say, in the reticulospinal tract, for example. So the spinal cord is organized. As we look around the spinal cord, obviously there are nerves which enter or leave the spinal cord, carrying both sensory and motor information. And so that's what we call a mixed nerve. A mixed nerve has sensory and motor information. So here is in green here, these are mixed nerves carrying both sensory information. These mixed spinal nerves will then split and send a branch to the back, known as the dorsal ramus. It is a mixed nerve with both sensory and motor information, and a ventral ramus, which goes to the front with both sensory and motor information. So because the dorsal ramus is mixed, we can feel our back and we can move our back. Because the ventral ramus is mixed, we can feel our back and move our back. So if the sensory neurons are in blue and the motor neurons are in red, both the dorsal ramus and the ventral ramus contain both. However, as the mixed nerves approach the spinal cord, they split to form roots, and these roots are not mixed. Only motor axons travel in the ventral root, where they will reach those anterior horns. If you recall, the anterior horns are where the soma of the motor neurons are. Only the sensory neurons will then travel through the dorsal root to reach the dorsal horns. And if you recall, the dorsal horns are where the soma are, which process sensory information. Now, while most neurons are multipolar, meaning that the soma is kind of at one end where the dendrites come out in one big axon, there are other shapes of neurons. Some are bipolar, in which they have two large processes come out. And so the soma is now more in the middle. And if a soma is now more in the middle, the question comes up, well, where do you put it? And the answer is here, what's called the dorsal root ganglion. So if this sensory neuron is a bit unusual in its shape, where the soma is in the middle, the question is, where do you put it? And the answer then is you would put it In a ganglion, and a ganglion is the name we have for a cluster of soma outside the brain or spinal cord. And because it's along the dorsal root, then it is called the dorsal root ganglion. So as you look at the spinal cord, there is a dorsal root which only carries sensory fibers. And then there is a ganglion there, the dorsal root ganglion, which has the soma of these sensory neurons. So the sensory neurons are bringing information in. Then there are what are called interneurons, or association neurons. They are neither sensory nor motor. Instead, they are going to process this information, to bring information together, to analyze it, to choose the best possible outcome. So most of the brain and spinal cord is composed of these interneurons, before then a motor neuron will then carry a command out to the periphery of the body. And so in a typical, say, a spinal reflex, you would have sensory information coming in from a receptor. And this sensory neuron could perhaps synapse directly with a motor neuron going out, but a more complex reflex would involve this sensory neuron then complexing with interneurons, which analyze the information before synapsing with the motor neuron, which would then carry the message to the periphery, giving a command, say, to muscles or glands. And so as far as the physiology and reflexes, I have more videos on that, but going to the anatomy. Getting back to those ventral rami, if you recall, a ventral ramus is a mixed nerve carrying sensory and motor information to the front. Well, there's something that is a bit confusing, because we could label these, we could call it C5, C6, C7, C8, T1. And if you ask what nerves innervate specific muscles, sometimes it is, like T6, T7 will go to some of the breathing muscles, and you say, oh, T6, I know how you got your name. You're leaving around the sixth thoracic segment, etc. But then a lot of nerves are called the radial nerve or the sciatic nerve. Where do those names come from? And that's because along the spinal cord, we see a number of these complex plexuses where the ventral rami, only the ventral rami, not the dorsal rami, the ventral rami of a number of different spinal nerves will then interweave. They kind of swap fibers. So if all of C5 is red, all of C6 is orange, all of C7 is blue, etc. Notice that some of the blue fibers end up going this way in this nerve. Some of the blue fibers end up going this way. And so when we look at the muscles of, say, the arm and chest, like the pectoralis or the deltoid or the triceps, none of them are innervated only by C7. None of them are innervated only by T1. Instead, because all of these fibers, then swap some of their axons, then these nerves, like the axillary nerve going to the armpit region, is going to have some C5, some C6 fibers. The radial nerve will have some of all of these. So this mixing, this interweaving is what's called a plexus, specifically the brachial plexus in this. And the nerves coming out of the brachial plexus then go to the muscles and the skin of the arm and chest region. So if you were going to memorize the innervation of these muscles, so like the radial nerve will innervate the triceps brachii, for example, the musculocutaneous would innervate the biceps brachii. You would then be memorizing the names of what comes out of the plexuses. So in addition to a brachial plexus, there's a cervical plexus here, which gets some of innervation from cervical neurons. And then there's a brachial plexus, which sends innervation to the arm and the and the shoulder. And then there is both a lumbar plexus and a sacral plexus, which will then send out muscles to the nerves to the hip and thigh. So things like the sciatic nerve, the femoral nerve, that doesn't leave the spinal cord. There's no femoral nerve coming out of the spinal cord. Instead, you have L1, L2, L3, or S1, S2. And so you would form a sacral plexus, a lumbar plexus, and then out of these plexuses come the nerves, which go to the individual muscles and the areas of the skin. And so I, have this little playlist on the anatomy of the human spinal cord. Just a couple final things. We find a swelling around the arm called the cervical enlargement. Just there's more processing and nerves coming in and going out to the arm. So the spinal cord's a little wider there. Likewise, there is a lumbar enlargement. The spinal cord does not reach all the way to the tip of the sacrum or coccyx. Instead, it ends here in the middle of the back, in the middle of, say, the lumbar vertebrae, at what's called the conus medullaris. And then from that point, there's a bunch of the nerves passing as what's called the horse's tail, or cauda equina, which then includes one small fiber that will go all the way to the tip of the coccyx, known as the filum terminale. So that's just some of the aspects of the anatomy of the human spinal cord. It is in a body cavity, the dorsal body cavity surrounded by cerebrospinal fluid. And also then there are meninges protecting it. So there's the dura mater, the arachnoid, and then there's a space underneath the arachnoid, followed then by the pia mater, which then touches the nervous tissue itself. Well, so with that in mind, that's the anatomy of the human spinal cord. How did this evolve? So then if we look then at the evolution of the spinal cord. There are invertebrates which have nerve cords, but in most invertebrates, the nerve cord is on the dorsal, I'm sorry, thinking of the heart. The nerve cord is on the ventral side of the body. So if you look at a crayfish, a worm, the nerve cord is on the ventral side, not the dorsal side as it is in us. And it's solid as opposed to what is a defining feature of the chordates, where the nerve cord is hollow and it's on the dorsal surface. So even if we look at primitive chordates like lancelets, we see that the nerve cord is dorsal along the back and hollow around a central canal. There are some features different from that of humans. One is there are some giant cells in the spinal cord of the lancelets, and also then in fish and amphibians, which would be lost in higher vertebrates. So we don't have these giant cells. And then also at first in the lancelets, while there is a concentration of soma around the central canal, the gray versus white matter is not clearly delineated. And so these are different. Another similarity is that the sensory axons are coming in the dorsal part of the spinal cord, and the motor axons are leaving from the ventral part. So once again, this is a similarity. So chordates, starting with even invertebrate chordates like this lancelet, they do have similarities. A difference is that the soma of the sensory neurons would be here in the dorsal region of the spinal cord, not in a separate dorsal root ganglion. And then also in the higher vertebrates like ourselves, the sensory information from the dorsal root will fuse with the motor information from the ventral root to form a mixed nerve. Whereas that doesn't happen here. There will be a sensory nerve which leaves the top of the spinal cord, and then a motor nerve that leaves the bottom of the spinal cord. If we were to then jump ahead to a lamprey, and here's a dissection of a lamprey spinal cord, notice that you can see a distinction between gray and white matter. The gray matter is on the interior, as in us. The white matter is outside of it. Here you see the central canal. So the spinal cord of a lamprey, a primitive vertebrate, is a little bit more like ours um, than the spinal cord of that lancelet. Uh, it's um, it's more of a solid tube than we see in lancelets, but it's a bit more flattened in shape. Um, the dorsal roots are more restricted to sensory information, whereas in chordates, there are some visceral efferents which lead through dorsal roots as well. Um, the dorsal and ventral roots fuse in hagfish, but not in lampreys. And so, because they are both jawless fish, what's the more primitive condition? Then isn't entirely clear. Now, spinal cords work and brains work a little bit differently in these jawless fish. So for the most part, the spinal cord's running the body. The brain that is running the head, and it doesn't send a whole lot of commands to the body. So most of the bodies then run on more local reflexes, not originating in the brain. But there are tracts which do reach the brain from the spinal cord, and there are homologs of tracts which we see in the higher vertebrates like ourselves. So there is a spinocerebellar tract carrying information about body's position to the cerebellum, which lampreys do possess, and a homolog of the tectospinal tract carrying commands from the midbrain down to the body. So even when we look at primitive vertebrates like these lampreys, we see homologs of some of the ascending and descending tracts, which we see in higher vertebrates like ourselves. If we now look at jawed fishes, the spinal cord is rounder, and the gray matter is more obviously divided into dorsal horns and ventral horns, just as we see in the human spinal cord. There are plexuses, although they are variable. And one of the big variations in the cartilaginous fish is rays. I mean, when you look at a shark is mostly swimming using an S-shaped motion with its body, whereas the rays are primarily using these broadened pectoral fins. And so given that, the innervation of these fins are different in sharks and rays, and so the nature of these plexuses obviously vary as well. Now, some of the ascending tracts which we see in sharks are homologs of ascending tracts which we see in humans, and the same would apply to some of the motor tracts, like the reticulospinal tract I had mentioned earlier. Once again, there's an organization here, and some tracts are myelinated, some are unmyelinated, as we will see in the higher vertebrates as well. And so when by the time of the first jawed vertebrates, spinal cords had a few more similarities to those of humans. When we look at the first bony fish, there are more similarities. The amount of gray matter increases. The dorsal and ventral roots are closer together and always fuse, which is not always the case in sharks. The complexity of synapses increases. The soma of the motor neurons are located more ventrally. And then we have a cervico-brachial plexus, dorsal and ventral fissures in the spinal cord, and a few more similarities. And so the spinal cord becomes progressively more like humans as we go from the first, primitive vertebrates to primitive jawed vertebrates to primitive bony vertebrates. Obviously, tetrapods, when they moved on to land and developed arms and legs, this then would increase the innervation of those regions. And so those cervical and lumbar enlargements, which I pointed out on the human spinal cord, they are present as well. The volume of the dorsal column for touch increases. The overall size of the spinal cord increases. So there's a cervical enlargement, for example, in humans. Um, there is both a cervico-brachial plexus, which later will get divided into two separate cervical and brachial plexuses, and then a lumbosacral plexus, which will form, once again, two separate plexuses, the lumbar plexus and sacral plexus in mammals. The spinal cord does not go all the way to the tip of the tail, and the final nerves form a cauda equina, a horse's tail, as we see in humans. There are more similarities in the tracks, such as from the red nucleus, a rubrospinal tract, which also exists in humans. By the time you get to early reptiles, dorsal and ventral roots always exit the spinal cord in the same segment, as opposed to alternating segments. Those giant cells of the spinal cord are no longer present, as they are in chordates, in fish, and in amphibians. The dorsal column for touch, if you recall, in humans is split into an area for the legs and an area for the arms, and that is true of all amniotes, the fasciculus gracilis for the legs, the fasciculus cuneatus for the arms. They would be the red and the yellow sections here. When we look at how the gray matter is divided, there are certainly differences when you compare, say, a reptile into a human, as far as the organization of the gray matter. But as you can see by the corresponding colors, there is homology there as well. And so as we're studying the spinal cord, the tetrapods and later the amniote reptiles, they have even more similarities to the human spinal cord than we saw in the fish and the amphibians. Finally, then, if you were to consider the mammals, this is where the spinal cord, as you can see in this cat, is very highly similar to that of the of humans. It gets shorter in its overall length. In the live-bearing mammals, it stops, the conus medullaris stops before reaching the sacral region. The spinal cord overall is bigger. And there's lots of things. The plexuses, as I had mentioned, they separate. So the cervical and brachial plexuses separate from a common ancestor, as do the lumbar and sacral plexuses. And a new tract, a very important descending tract called the corticospinal, coming from the motor cortex of the cerebrum of the brain, which will allow voluntary command of skeletal muscles. So this exists. It doesn't exist to the degree that it will exist in primates. So the corticospinal, it doesn't proceed very far down the spinal cord at first, but mammals have this corticospinal tract going from the motor cortex down the spinal cord. And then in primates, it becomes much more highly developed, allowing for, in the hands, for example, great manual dexterity here. So primate spinal cords increase the size of the cervical region, the corticospinal tract larger and more complex. And then we can see some additional changes as we get to the catarrhine primates, those are the old world monkeys and apes and others. And so once again, as we look at the spinal cord, obviously, whether it be the digital dexterity in the hands of primates or the coordination of running, these are all using anatomical aspects of the spinal cord, which have evolved slowly over time. I end this then with pictures of the spinal cord of the lancelet, Amphioxus. There's the notochord. Here's the spinal cord. Over it, it is hollow. There is a central canal, but no obvious gray and white matter, as you can see. So there's the notochord here. Here is the spinal cord above it. It is hollow. And so, I just go through some of the aspects of the lancelet. a spinal cord here. A couple of quick comments. The dorsal root cells are in the spinal cord, once again, not in a separate dorsal root ganglion. There are reflexes which stay on one side of the body, but they can also cross to the opposite side of the body. So there are commissural cells which are on the midline and can help information cross. Here's that central canal. Once again, they're hollow. As far as the motor neurons, there are different ones. Some are going to skeletal muscle and are called somatomotor tracts. Some are going to other systems like the digestive system or smooth muscle and are called visceral motor tracts. As reviewed here, there are those giant cells, including some which are then involved in vision. So lancelets, they don't really have a good eye, but instead they have cup-shaped sensory cells known as organs of Hess, where a photoreceptor contacts a cup-shaped pigment cells. So it's actually the brain and spinal cord which are detecting light. So if that's the notochord, here's the spinal cord there. So that, I have a video explaining some of the features of the Amphioxus spinal cord. And then finally, another video just reviewing
the evolution of amphibians, such as the frogs.
Reviewing some of the features of the amphibians, which I had mentioned. So here's a frog spinal cord. And as you can see in the video in just a second, you can divide it into the gray matter versus the white matter easily. The gray matter is here around the central canal. There are fissures, a dorsal fissure and a ventral fissure. There's the fissure there. These were traits in higher vertebrates. You can make out where nerves are leaving the spinal cord. So here's the root of the root of a spinal nerve leaving. And so this was just a quick overview of the anatomical structures of the human spinal cord and also the evolution of these features looking at the spinal cords in diverse vertebrates.